So now for today's lecture, we would be starting with that how come the urine formation would take place in our body, and that is an important role that is rendered by the excretory system, or in other words, we say that the urine would be produced in the kidneys and then ultimately from the ureter and the urinary bladder and the urethra off over this system, so this would go out of the body. So the urine formation basically happens with the help of the two major processes, and these two major processes they are the ultrafiltration process, and the second one would be referred to as the selective reabsorption. So before I take you into the ultrafiltration or the selective reabsorption phenomenon, we must be clear about that how come these waste materials that we have produced at every part of the body, so they would be carried to the kidney. So the answer is very simple that wherever the waste materials they are produced, like in the cells, we have produced the CO2 and the metabolic water, they would go into the blood plasma. From the liver, the urea would go into the blood plasma. From the muscles, the creatinine would go into the blood plasma. And from the breakdown of the nitrogenous bases of the DNA, the uric acid is coming over here. And from the diet, the excess water and the ions that we are taking up, the vitamins and the minerals as well, so they are gone, or they would be there in the blood plasma. Now, from here, the blood would go to the heart. From there, it would go to the lungs with the help of the pulmonary artery, where we would do the gaseous exchange and we would remove the carbon dioxide. And also, some amount of the water would also be removed. And this oxygenated blood would be returned with the help of the pulmonary vein. The blood always oxygenated, but this has got a lot of waste materials into it. Now, from the heart, the aorta would go out, and the aorta would give the blood supply to the renal artery, and from the renal artery we would have these afferent arterioles coming. These afferent arterioles would take the blood supply into the glomerular blood capillaries, and from there these glomerular capillaries we would do the ultrafiltration process, where some components of the blood plasma would be taken out here into the Bowman's capsule, and when the blood would be cleaned up from here, so ultimately we would get rid of the waste materials. Now, if I were to quickly summarize this, so in the blood plasma, this is actually the yellowish liquid part of the blood. It has water and the nutrients and the waste materials, and among them the CO2 we removed it into the in the gaseous exchange thing, and we have still got this urea, uric acid, and creatinine. We also have these hormones within the blood plasma that can be the insulin and the glucagon and the antidiuretic hormone, and there would be the presence of these antibodies as well. There would be the minerals in this, the vitamins in the blood plasma, and the blood plasma would also have these clotting proteins in them. So this means that this blood plasma has got the waste material. So out of the blood plasma, we need to filter them out into the Bowman's capsule by the process of ultrafiltration. So if I were to tell you the definition of the ultrafiltration, so we would say that this is the fundamental process in which some of the constituents, not all the blood plasma, but some of the constituents or the component of the blood plasma, so they would be filtering out of the glomerular capillaries, and finally they would come into the empty space that we have in the Bowman's capsule. And over there we would make a filtrate, and that filtrate is the glomerular filtrate, we say. Like what I mean with this is that the first part of the nephron, you see that this was referred to as the Bowman's capsule, a U-shaped structure, and within the Bowman's capsule, now you know that here we have these glomerular capillaries coming, and these glomerular capillaries they were taking the blood supply out of the renal artery with the help of this afferent arteriole, and then on the other side of the glomerular capillaries we have the exiting arteriole, which was the efferent arteriole with me. So when I would take some constituent out of here in the blood plasma, and then this would come into the lumen of this Bowman's capsule. So this is what we refer to this as an ultrafiltration, we say. Now the scenario is that within these glomerular capillaries, I am required to be generating a very high pressure, and that high pressure is high hydrostatic pressure that would actually compel the waste materials that are present here within the blood plasma of the glomerular capillaries, so that it should be pushed out of here and it should come here in the lumen of the Bowman's capsule, and over there we would make the glomerular filtrate out of it. Why we call this as a glomerular filtrate? Because this is something that we have taken out of the glomerular capillary. So this is why this is the glomerular filtrate with us. So my idea would be that how come I would be able to generate a high hydrostatic pressure, that is a requirement of the ultrafiltration process. So if you were to see this diagram, so I have just taken the first part of the you could say nephron, because in the nephron we have this Bowman's capsule, and when we have got these proximal convoluted tubule and the loop of the Henle, the DCT, and then at the end we have got these collecting ducts with us. So we must know that this ultrafiltration is happening here. So if you were to quickly see this diagram, so this is the Bowman's capsule. Here we have the glomerular capillaries. Here I have got the afferent arteriole, and then here we have the efferent arteriole. So how I am generating the pressure? The reason is that the afferent arteriole, which is carrying the blood into the glomerular capillaries, so this has got a wider diameter in it. And on the opposite side of the glomerular capillary is the efferent arteriole that I have with me. So this has a narrower diameter of it. So this basically suggests is that the amount of the blood that enters from this afferent arteriole, it cannot escape out of the efferent arteriole at the same rate. So if we have got more blood coming from here, and if we have got less blood flowing out of here, so this would generate a very high hydrostatic pressure within the glomerular capillaries. And under the impact of that high hydrostatic pressure, what would happen that the urea that we have in the blood plasma, the glucose that we have in the blood plasma, the salts that we have in the blood plasma, and the water that we have in the blood plasma, so they would filter out of here. And when they would come into the Bowman's capsule, so here we are making the glomerular filtrate. Okay, so if I were to be describing this process for you, that how this ultrafiltration situation is done, so we would say that the afferent arteriole, it has a wider diameter and more blood would flow out of it. The efferent arteriole that we have here, so this has a narrower diameter, so the less amount of the blood would get out of here. So this would mean that the amount of blood that enter from here, it cannot escape from here. So as a result of that, we create a high hydrostatic pressure within the capillaries, and these capillaries are the glomerular capillaries, and this high hydrostatic pressure would push the constituents of the blood plasma, and they would come in out of the glomerular capillaries into the Bowman's capsule, and over there we have made a filtrate out, and that filtrate is referred to as the glomerular filtrate. So we must be now very much clear that what get filters out in the ultrafiltration. So we say that most of the urea and the uric acid and the creatinine and the minerals and the vitamins and the water that we have in the blood plasma, so they have got filtered out. And the problem is that all of the glucose and the amino acid, they would also be filtered out of the glomerular capillaries, but in the blood plasma, the red blood cells that we have suspended in it, and the high molecular proteins like the clotting proteins, the prothrombin and the fibrinogen and the stuff like that, so they are not filtered out. The reason is that these have got a high molecular mass of them, and since they have the high molecular mass, so that is why they cannot get out of the glomerular capillaries. So they would be held back in the blood plasma. Now here we have got a little problem as well, that this removal of the urea, the uric acid and creatinine and excess mineral and the vitamin and the water is something important, but the glucose and the amino acid that are getting out of the glomerular capillary, now this is something not good because they are the normal need of my body cells. So I would not allow them to be removed in the urine. So we are required to be reabsorbing them back, and that would happen in the second step of the urine formation. And we were calling this as the selective reabsorption. So for the next class, we will see that how come my body would take this glucose and the amino acid and some of the minerals and the vitamins and the water back into the blood by the ultrafiltration process. And then that would actually be making the urine at the end. So see you for the next class.